That's the soccer team that it's really good. Be here for a little while. Long enough that you'll be back.
Alright. So last whole week of class is this week. We're almost there, almost to the end of the semester. Hopefully you've been studying for the final exam. Because I know you have a lot of finals and probably final projects due at the end of the semester, so always better to start studying early. Especially those cumulative questions, right? Because that's almost guaranteed 20 points if you study those. Content check wise, we have one more today, we have one on Thursday, and that's it for content checks. There is that eugenics assignment also due tonight if you haven't done that already. Watching a couple of short videos. Otherwise, we're getting to the end of the semester here. So today, we actually have more of a math leaning lecture, which I know is not everybody's favorite. But this is the only math that we're really going to do this rest of the semester. In fact, I have to cut information out of some of our next lectures because we just don't have enough time. And I'm likely going to cut out some of the calculations. So that's good news for you. On that note, the study guide that I posted for the unit, whatever we don't get to, I will go in and cross off those questions so you know that there are questions that we did not cover in lecture, and thus you would not have to know. All right, but like I said, we're going to have kind of a math-heavy lecture today, so let's jump right in. So again, we're talking about the population. So just as a review to what a population is, right? It's a subset of individuals of a particular species that occupies one geographical area and has the ability to interact and thus reproduce with one another. Right, so we gave the example last time we talked about a definition of a population of these Joshua trees. Right? So it has to be a group of sexually reproducing organisms. Right? If they're an asexual species, they would have no need to interact with one another to reproduce. Right? Thus, would not fall into this category. Right? So that's sexually reproducing organisms. They have the ability to interbreed, which is oftentimes what defines a species, the ability to interbreed with one another. And they're in the same geographical location, right? So there's easy traveling distance between individuals. They all have the opportunity to interact. Right? Now, if we think about organisms, this can vary quite widely. Right? If we think about mice, right, their geographical range might be confined to maybe like one farmstead or one house or one area, right? Versus humans, right? Theoretically, you have the ability to interact with almost every single human on Earth, right? Now, it's going to be limited by travel and time and expenses, right? But theoretically, we have a very large population range. Right? You think about, say, wolves, for example, right? They have large territories. They cover a lot of space. Their range is quite large compared to mice or other smaller organisms. Right? So a population, right? The area in which they occupy can vary depending on what animal, what organism we're talking about. All right, so within a population then, we could look at all of the genes present. Right? We would call this the gene pool. Right? All of the alleles for every gene in a given population. Right, now, a lot of times we don't look at the entire gene pool. We might look at just one or two alleles at a time. Right? But the gene pool applies to every single allele present for every single gene in these organisms. Right? So if we're looking at population genetics, which is what we're going to be focusing on today, right? We're going to look at the variation within that gene pool, and how much diversity is there. We could also look at how that gene pool might change from one generation to the next. We're not going to get to those equations in this semester. But if you take, say, evolution, ecology, and behavior, you will get to those types of equations. Okay. Like I said though, whenever we look at gene pool, the emphasis is often only on one gene at a time, or one set of alleles at a time. So if we look at this population of ladybugs here, right, we can see that their genotypes have been listed. Right? There's 3 different phenotypes, 2 different alleles, right? 3 different genotypes. Right? And so theoretically, I could go and look at the gene pool for this particular allele and determine how frequent is it that the individuals in this population will be heterozygous, or how frequent is the dominant allele going to appear in this population. All right, so we're looking at genes, right? This is not new content, but this is maybe new terminology. Okay? Genes are usually going to be polymorphic, meaning there's going to usually be more than one allele for a gene. Right? We've seen that, right? We've talked about a lot of genes like capital A and little a, right? That's 2 different alleles for one gene. Right? There are instances where a gene might be monomorphic, right? It's much more rare, right? So for example, these birds, they're monomorphic in the plumage gene, meaning what determines how their plumage looks, or their feathers, is only controlled by one allele. And there's no variation in that allele. All of them look the same in terms of plumage, right? But most of the time they're polymorphic, right? 2 or more alleles. Right? And a lot of times these polymorphic genes arise, so we get variation in our genes due to point mutations. Right? Or we talked about maybe a sense or a missense. Right? So those point mutations, if they're not going to lead to the organism not surviving, if the organism survives with it, the mutation could be beneficial. Right? It could be neutral. But that mutation might change the variation within the alleles of this population. All right, so when we are looking at population genetics, we are going to be looking at polymorphic genes, right, where they have at least 2 alleles. And in fact, for this lecture, we're only going to look at 2 alleles at a time, right? We're going to keep it simple for ourselves. All right, so if we have a population, right, how are we going to analyze this genetic variation? Right? What would we might want to know about this population? Right? Well, we want to know frequencies. We wouldn't want to know allele frequencies. So how often is there going to be a dominant allele or a recessive allele? But we'd also want to know genotype frequencies. So how often is it that these individuals are going to be heterozygous, or homozygous dominant, or homozygous recessive? Right? How often is this going to be the case? Right? Because we're interested in genetic diversity in these populations. We want to know what the genetics of this population looks like. Right? We want to determine these frequencies. All right, so we're looking at a population that is polymorphic for their genes. We're looking at one gene at a time within the entire gene pool, and we want to determine how frequent an allele or a genotype is going to show up. This is where we start to get into the math. All right, so we can determine allele frequency. And we can determine allele frequency by looking at the number of copies of a specific allele in a population divided by the total number of all alleles for that gene in a population. So if I have this population, right, in this green circle, I've only listed the instances of a single allele, right? Say I want to know how many times a recessive allele might show up in this population, right? What is the allele frequency for the recessive allele? I would count out the number of copies of that recessive allele. So in this case, 1, 2, 3. right? So I put 3 on the top, divided by all of the alleles for that gene in the population. So that would be 1, 2, 3, 4, 5, 6, 7, 8, 9. Right? So I get 3 divided by 9. Right? If I did the math, I would come up with a decimal number which I could change to a percentage, right? And that would be my allele frequency. I could do the same for genotypes. So rather than looking at one allele at a time, now we're looking at the combination of 2 alleles. Right? And I noticed this on my last lecture that one of my genotypes is actually missing a second allele. There should be a second allele there. All right, but say I want to know the frequency of individuals in this population being heterozygous. Right, so a capital and a lowercase letter. Again, I would count the number of individuals with that genotype I'm interested in. So heterozygous individuals, that would be 1, 2, 3, 4 individuals, divided by all of the individuals in the population. So 4 divided by 1, 2, 3, 4, 5, 6, 7, 8, 9. Right, so 4 divided by 9, that would give us a decimal. We could turn that into a percentage, and that would be the frequency of individuals in that population being heterozygous. All right, so fairly simple equations at this point. Let's put these equations into practice. So we're going to look at this plant, right? We call them 4 o'clock plants. That's the common name for these plants. And this plant has 2 different alleles, right? And 3 different phenotypes. Why might it have 2 alleles but 3 phenotypes? What type of situation is occurring here? Not codominance, but incomplete dominance, where it's a blended phenotype. Yeah. So this one has that blended phenotype of having a pink flower. All right. We're looking at a population of 100 of these plants. Based on their flower color, I can determine their genotype. Right? If they are red, they would have the genotype of RR. Right? And I know 49 of those individuals have the RR genotype. 42 are pink, so they are heterozygous individuals, and 9 of them are white, so they're homozygous for the white allele. All right, so I know these values, and I know them just by looking at the color because I know they are exhibiting incomplete dominance. All right, so remember homozygous versus heterozygous. I'm going to say these words a lot today, right? Heterozygous means a copy of each variation. Homozygous means you still have 2 alleles, but they're the same allele. So I want to calculate frequencies, right? Based on that population where I know how many of each plant exists, and thus know their genotypes. I want to look at the allele frequency. I want to see how frequent is it that this white allele shows up in the population. All right, so I'm going to look and count the number of times I might see the white allele divided by the total number of alleles in the population. Now this is where things become just a little bit more complicated, right? So we're looking at the white allele. There are 2 genotypes that contain the white allele, right? Either they're a homozygous individual for the white allele, so they contain 2 copies of the white allele, or they are heterozygous and contain 1 copy of the white allele, right? So if I look at the total number of the white alleles, I would have to take the number of individuals that are heterozygous, and thus contain 1 copy of the white allele, plus the number of individuals that are homozygous for the white allele, right, so WW, and multiply that by 2, because there's 2 white alleles in that one genotype. All right, so now I would plug in my numbers, right? I have 42 heterozygous individuals. I have 9 plants that are white. I'm multiplying that 9 by 2 because there's 2 of those white alleles in that genotype. And I'm dividing it by all the alleles in the population. So each genotype has 2 alleles. Therefore, we have to multiply each genotype by 2 to get the total number of alleles. Right, now if I do that and I simplify everything, it will come down to 60 divided by 200, or 30%, 0.3. All right, so the big thing with the alleles is that we have to multiply for the homozygous individuals for the allele that we are interested in. All right, we can do the same thing for genotype, and it's actually a little easier, because now we're looking at genotype as a whole instead of individual alleles. And I want to know the frequency of having the genotype WW in my population. I would have to count out the number of individuals that have that genotype, and I know the number of individuals is 9, divided by all the individuals in a population, which if I added all these individuals up, it would be 100. So I know 9% of these flowers are going to have the genotype of WW. All right, best way to learn is to practice. So I want to know the allelic frequency of having the red allele, or the capital R. This example up here is the example we just went through with the white allele, so you will have to alter this example for the red allele and come up with a percentage. And I'll give you a few minutes here.
Yellow box, you guys know the name out. Those are the specific so there's 49. Multiplied by 2, so yeah.
These are all multiplied by 2 because we know for the phenotype that 2 alleles.
I might just like think that it's not that.
Oh, okay. Okay, do you think that one?
If that's what you guys got, then it's just the same.
She wants the frequency of the CR.
Like,
So like add in a lot. Or allele. How do you feel about the I'm not worried.
yeah.
I think it's mostly release and stuff. Which makes it easier for me. You know your code when you're like in a test and you're like, how many It is a habit though. I do it now. It's out of habit. I'm like, how many of these do I think I can work with? 70 last night. And then well, I'm like, how many of these are like uncertified? No. How many of these do I think how many of these do I think I got 100% right? And then I add the uncertainty, the wrong, and then I add 5 more in case I forgot about the other questions, and that's the worst. Yeah. So I like,
Oh.
I very good. So then you never just
Yeah.
Alright, so if I'm trying to find the frequency of this red allele in my population of 4 o'clock plants, how am I going to set up the top part of this equation? What will it read? Alright, let me ask a different question. What did we get for an answer?
70%.
Yeah, 70%. Right? So, 70%. And so how do we set up the top line here? What values did we add and multiply together?
42 and 49.
42 plus
2 times
2 times 9. Yeah. And so we got 140 at the top, right?
Yeah.
Oh, excuse me, yes. Yes, I was looking at my W example. Right? 2 times 49. Alright, so we should have something like what I have up on the screen here, up at the top. Right? 42 plus 2 times 49, not 2 times 9. Right? And it boiled down to 140 over 200.
Are we
Which
gonna
equals
go on, like,
0.7 or 70%. What was that?
Get calculators on the test? Because I can't do that.
I am still trying to decide how I'm going to do this. Normally, I would give you calculators on the exam, but I'm going to have 70 students taking this at the same time. I don't think I have 70 calculators to be used at the same time.
So we're not gonna be tested on it.
Can we do fractions?
Because I can't do that.
You could do fractions. So I think what's going to happen is I might have you set up the equation, but not actually calculate the equation out.
Oh, okay.
Yeah, I think that's what I'm going to do. I still have to look. It might be that I can find 70 calculators.
You
So
could make it
this
extra.
will be on the exam. In what format, I have to figure out still. All right, did anybody find 70% in a different way than using this equation? There's an easier way to do this.
The other side is 30%. So
Yeah.
that's what they said.
Yeah, we already calculated out that the frequency of having the W allele is 30%. And when there's only 2 alleles in the population,
We're all done.
so 100% minus 30% would be 70%. I purposely did not tell you how to do this because I wanted you to do it the more complicated way, right? But there was an easier way to do this. All right. Let's keep going and expand upon our population genetics. We're going to learn a couple of more of equations here, all right? And we're going to learn what's called the Hardy-Weinburg equilibriums, right? So an equilibrium equation in this sense is looking at the genetics in a population, but it's a population that's not currently evolving, right? So it's going to predict the allelic frequencies. It's going to predict the genotypic frequencies, but this is in a population where forces of evolution are not at play. Right? So that means there's going to be no new mutations occurring. Right? So natural selection is not going to occur. Right? Mutations and natural selection are both mechanisms by which evolution can occur. The population is going to be relatively large, meaning genetic drift is not acting upon this population. And finally, there's going to be no migration between different populations. Thereby, there's no gene flow that could allow this population to evolve. Right? And theoretically, there's more or less random mating. Right? So this population is not evolving. In reality, this is hardly ever the case. Right? We're rarely going to see a population where evolution or changes in allelic frequencies are not at play. All right, so why do we care about this equation? Well, basically what this equation does is give us a snapshot into the population. We can look at a population at a point in time. What we cannot do is look at this population across many generations, because rarely are we going to have multi-generations of a population in a population that's not evolving. Right? But we can look at a snapshot in time. Right? And if we look at a snapshot in time, and we might look at a couple of different snapshots in time, and if the values we get stay the same, right? It means evolution is not occurring. But if we compare these different snapshots in time and the values do change, that would be an indication that there's some evolutionary mechanism at work, right? So basically we're using these equilibriums to understand what this population looks like at this point in time. We're not using it to predict how it's going to change. We're not using it to calculate how the populations change over time. We're just looking at it when I went out and I did a census of butterflies on April 2nd at 5 o'clock in the evening in the year 2025. Just a snapshot in time. All right, so our Hardy-Weinburg equilibrium. Population is not evolving. There are two different equations that we can use. Right? One equation is to determine the allele frequencies, and one equation is to determine the genotype frequencies. Right? So we're still determining the same thing that we used those equations we just talked about. Right? We're still determining allele frequencies and genotypic frequencies. Right? We're just going about it in a different way. All right, so allele frequencies would be P plus Q equals 1. And the genotypic frequencies would be P squared plus 2PQ plus Q squared equals 1. All right, that probably means nothing at this point, right? The P's and the Q's are just letters that are put there to represent the versions of the alleles. I don't know why they decided to use P and Q, right? And not T and V, right? Why not another set of letters? I don't know. But what I do know is P is going to represent one allele, so maybe it's the capital letter, and Q is going to represent the other allele, so the lowercase letter. So if I have an individual that is a heterozygous individual, so it has a capital and a lowercase letter here, a dominant and a recessive allele, I could assign P to the dominant allele and Q to be the lowercase allele. Right? You could have flip-flopped it and said Q is now the dominant allele and vice versa. Right? It really doesn't matter as long as P and Q are representing different alleles. All right. So again, let's put this into practice. Right? We're going to use our same population of 4 o'clock plants. Right? And we know their allelic frequencies. Right? We just calculated out the frequency of there being the red allele, which we calculated to be 70%, and the frequency of the white allele, which we calculated to be 30%. Right? We know these percentages. If we know these percentages, we can simply plug them into an equation to find the genotypic frequencies. All right, so we're going to say P is going to represent the red allele, and Q is going to represent the white allele. I have my equation, P squared plus 2PQ plus Q squared equals 1. This is an equation that, yes, you should memorize. So I'm going to plug in the values here. P squared, right, P is the red allele. The red allele has the frequency of 0.7. So I can plug in 0.7 squared and get the frequency of my homozygous individual for the red allele, RR. 2PQ is going to be the frequency of the heterozygous individual. So I would go 2 times the value for P, so P is R, which is 0.7, times Q, which is the value for W, which is 0.3. And I would get the frequency of my heterozygous individuals. And then Q squared would be for my homozygous individual for the white allele. Right? So Q is W, W is 0.3. I am squaring that value, and that would give me the genotypic frequency. Right? So if I plug in those numbers, I would get a frequency of 0.49 for my homozygous for my red allele. My heterozygous individuals would be 0.42, and my homozygous individuals for the white allele would be 0.9. Right? Or if you change them to percentages, you could say 49%, 42%, and 9%. Now these numbers should ring a bell, right? Because we actually already knew the frequencies of these genotypes, because I told you how many individuals had the particular genotypes in the previous example. Right? So what this means, right, is we could apply this to a Punnett square to figure out the frequency of offspring having particular genotypes. Right, so I'm going to actually go to the next slide to explain this. All right, if we have two parents, right, we have a parent that is heterozygous, so has an R allele and a white allele. The other parent is heterozygous, has an R allele, has a white allele. Right? If I know the frequencies of my alleles, right, and I know the red allele has the frequency of 0.7, right, in this Punnett square, the genotype of RR is likely going to occur in the offspring, 0.49%. Right, the probability of these parents making a heterozygous zygote is 0.7 times 0.3, which would be 21%. But there's two instances of a heterozygote, right? So it's actually 0.42%. And we could do the same for the white flower, right? 0.3 times 0.3, there's a 9% chance the offspring will have this particular allele, right? So far we've done Punnett squares where we haven't known the percentages behind them, so we can predict genotypes. But now if we know allelic percentages, we can predict with a relatively certain degree of certainty what the genotypes, how often the genotypes will occur, right? Rather than just what the genotypes will be, right? Now it's important to note that these genotypic frequencies must equal 1 or really close to 1. Sometimes rounding errors might give you like a 1.02, right? So if you went through and did this equation and these values did not equal 1, that would be an indication that something went wrong. You should go back and check your work. All right, again, this population, no evolution is occurring. That is key. I want to emphasize that here. No evolution is occurring. This is very rare to actually happen in nature. We're looking at just a particular snapshot in time. All right, let's go through a more complicated problem together. A problem that is more likely to show up on an exam, right? So, we're going to look at a problem where we're going to analyze genetic variation in our population of deer, right? We are going to look for the allele frequency and the genotype frequencies. And we're going to use our two Hardy-Weinburg equilibrium equations. And we are only going to start with a small amount of information, right? I am only going to tell you that there's a population of deer, right? There's 200 individuals of deer in this population. 65 of them are white, and white is the recessive allele, right? Just from this information alone, and applying our equilibrium equations, we'll be able to figure out everything we need to know. Right, but we're going to take this step by step. So what do we know about this population? Right, we know there's 200 deer. We know 65 of them are white. And the white deer, or the white allele, is the recessive allele. So we can figure out genotypes. Right? What would the genotype of this white deer have to be? Yeah, both little case letters. And I'm using A's in this case. Right? So it'd be little a, little a. We know it has to be homozygous recessive. Right? What about the brown deer? What could be the possible genotypes here? We can't say one for certain. We know it has to be either homozygous dominant or heterozygous. Right? But we can't say for certainty which it is. All right, so we know basically the genotypic frequency of homozygous recessive individuals of white deer in this population. Right, so we're going to use our Hardy-Weinburg equilibrium equations, both the allele and the genotype frequencies. we're going to say that our P equals our capital A and our Q equals our lowercase a. We know 65 of the 200 of the deer are white, and we know that those white deer have the genotype of little a, little a. Right? If we did the math, 65 divided by 200, we would know that 0.33 would be the frequency of having a homozygous recessive individual in this population. Right? Or 33% of this population should carry the genotype of little a, little a. All right, so we know this information just by those two values that we were initially given. 65 of the 200 deer were white. All right, so we know the genotypic frequency of little a, little a. Great, we're going to use this. Right, we're going to take this information and start applying it to our other equations. All right, so we have our two equations. We are going to use the genotypic frequency equation first because we know the genotypic frequency of the little a genotype. Right? So we could say capital A squared plus 2 times capital A times lowercase a plus, right, 0.33 squared. Because little a squared, we know little a is 0.33. All right, so we would plug in our 0.33 where our genotype would be for little a, little a. Right, now that doesn't tell us much on its own, but we can do some simple math to now figure out, instead of just the genotype frequency of little a, what would be the frequency of just having a singular a? In order to do this, we actually have to take the square root of 0.33, right? Because it was initially squared in our equation. We're going to take the square root of 0.33 to find an instance of just a singular a, which would then divide out, or square root out, to be 0.57. All right, so now we know the frequency of the little a allele. Yes?
So this 65 out of 200 is Q squared.
Yes. Yep. And we know that only genotype for white deer would have to be little a, little a. All right, so now we know the allelic frequency of a little a. And we got to this allelic frequency by using the genotypic frequency of little a, little a. All right, so now we're going to use what we know about our little a. We know the allelic frequency is 0.57. We're going to plug it into this top equation here. P plus Q equals 1. Right, so I would say P plus 0.57 equals 1. Right, if I solve for P, I would take 1 minus 0.57. So then P has to equal 0.43. We know P is the capital letter, the dominant allele. So the allelic frequency of the dominant allele would be 0.43. All right, so now we know the allelic frequencies for both the capital and the lowercase a's. And we know the genotypic frequency of little a, little a. Once we've gotten to this point, right, now the rest of it becomes just you plug in the numbers into the equation and you solve the equation. Right, so we know capital A is 0.43, little a is 0.57. I'm going to plug in the numbers into my equation here, p squared plus 2pq plus q squared. Right, so if I plug them in, I solve the equation. So 0.43 squared is p squared. Right, 0.43 squared equals 0.19. P squared, right, stands for capital A, capital A. So my genotypic frequency of having 2 capital A's is 19%. Right? I could do the same for my heterozygous individuals. I would take 0.43 times 0.57 and then multiply that by 2 to get 0.5. So 50% of my individuals are heterozygous. And I already knew this, right? I already knew that 33% was little a, but I could plug the numbers into the equation. So I could go 0.57 squared to get 33%. Now if you add all these values up, they should be close to 1. Now I think if you actually add all these values up, you get 1.02, which is just has to do with the rounding. So likely, I rounded this 0.33. It was likely 0.33333333, all right? So as long as it's really close to 1, you're fine. If it's 1.3 rather than 1.03, then I would go back and check your work. All right, so I now know my genotypic frequencies of all of my deer. Right? So I have solved this entire problem, and I was only given 65 deer were white out of a population of 200. Right? I figured out the allele frequencies, and I figured out the genotypic frequencies. Any questions? All right, best way to learn is to practice. All right, I didn't know if we'd get further than this today, so I put the homework slide in the middle of the lecture, but I do have a few more slides to talk about. All right, so we've been talking about a lot about populations, right, which is a single species. Now we're going to start talking about how do we determine multiple species, or what makes a species a singular species. All right, so there are some terms that you'll need to know in order to understand how a species becomes a species. All right, the first two terms kind of go hand in hand, right? Microevolution and macroevolution. So far, when we've been talking about evolution, we've really been talking a lot about microevolution, which is the change in the allelic frequencies in a singular population. Right? If we were to talk about macroevolution, this is where we talk about evolutionary changes that create a new species. Right? So far we've been talking about how populations change and evolve. Right? But if we were to talk about how species arise, we would be talking macroevolution. All right? We also need to know the difference between species and speciation. Right? So a species is a group of organisms that share a particular set of attributes. Right? That's about as specific as we can be in a definition for a species, because we will see those attributes that they share can vary based on how we might determine if they're a different species or not. Right? But different sets of attributes might be how they use their environment, what they look like. It might pertain to their DNA, how similar their DNA is. There's a wide variety of attributes that we can look at. And then the word speciation is just how new species arise, the formation of a new species. Right? And again, speciation is macroevolution, right, that has occurred and likely has occurred by the accumulation of many different microevolutions within a population or within a species. All right, so we got macro versus microevolution. We got species versus speciation. So we're going to talk about how do we determine what is a species, right? What makes a species a species? How can I look outside and say that that bird species is different from the bird species on the other tree, right? How would I make that determination? So when we're distinguishing between species, most of the time, we're looking at sexually reproducing species, and we're determining if a species can reproduce with another individual or not. Basically, if two individuals are able to mate and produce offspring that is viable, we would consider them the same species. But if if they try to mate and they can't, or they mate and they produce offspring that isn't viable, maybe the offspring is sterile, right? Or maybe the offspring dies right away, we would consider them separate species. Most of the time that's the definition we go off of, but that is not the only definition we can use, right? There are other common characteristics, like morphological features, how they look, right? The ability to interbreed, so reproductive success. We can look at their DNA, their molecular features, as well as how they use their environment, so like the ecology around them. And it really depends on the species that we're talking about, what characteristic we are going to use to tell them apart. All right, so for example, on this image here, I have several different species of a single-celled organism. Right? If I were to use how they look to tell them apart, I would probably have a really hard time because they all look really similar to each other. That might not be the best characteristic to use here. We might have to use something else. So let's talk about these common characteristics. So the first one we're going to talk about are morphological traits. So the physical characteristics of an organism. What does an organism look like? All right, so on this screen I have given you a picture of 4 different frogs. How many species do you think are depicted between these 4 different frogs? What would you guess? You have 3, 4, anyone guess 2? Okay. It's hard to tell, right? We're using just how they look to tell them apart. So we're looking at their morphological features. On the screen, right, I actually have 3 different species, right? So the species up on top, the blue and the yellow frog, are actually the same species, right? If I was using morphological characteristics, I might say that these are different species because they look different, right? But that's actually the same species. The bottom row is also a same species, or different species, but they look the same, right? So these are 2 different species, but if I were to use physical characteristics to tell them apart, I might say that they're the same species because they look similar, right? So morphological traits are great because you just have to look at an organism to tell them apart, but there's drawbacks, right? So how many traits might you consider? Right? Am I just going to look at their skin color? Or am I also going to take into account how many spots they might have on their skin? Or am I going to use a combination of 3 traits or 4 traits to determine if they're a different species? Right? Traits within a singular species can vary, like these frogs up at the top here. They're the same species, but their skin color varies. So varies from yellow to blue. Maybe they're all blue and they're a darker blue and they're a lighter blue, right? There's a continuation of traits. And then finally, what degree do they have to be dissimilar in order for them to be separate species, right? Think about these bottom species here, right? They look the same. If you looked closely, you could start to see some instances where they are dissimilar, right? But they look mostly the same. Right, so how different do they have to be in order for you to consider them a different species? Right, and again, members of the same species can look very different, right? And members of different species can look very similar. So there are drawbacks to this method. Right, we could use reproductive success or reproductive isolation, right, different means of how species might breed with one another to determine if they're a different species. Right? Reproductive isolation refers to them physically not being able to mate with one another because there's some sort of a barrier. Right? That barrier might be physical. Like in my image up here, I have 2 foxes. Right? And they have a physical barrier of this river that they can't swim across. They're isolated reproductively. So even though maybe they're a fox species, maybe they're 2 different species of foxes because they don't have the ability to interbreed. Again, there's drawbacks here, right? This might be easy to physically observe. We watch a population over time in their environment and determine that they can't interact with one another. right? But the drawbacks might be that it really might be difficult to determine in nature, right? Unless you are living in that population 24/7 and you know the ins and outs of that population, I can guarantee there's something about the population that you are not observing. Maybe, right, we're looking at these foxes, and we went out for 4 different observation periods of 2 hours each, right? And when we observed them, we noticed that they couldn't swim across the river. But maybe somewhere upstream, the river got shallow, and maybe they're actually crossing the river there, and we just didn't observe it, right? Maybe there's no geological barrier. Maybe they live in the same area, so they have the potential to breed with one another, but they simply don't, for a variety of reasons, right? They don't interbreed, right? Kind of a similar example might be if you're trying to breed animals in captivity. A lot of times you might put the same species in an enclosure, and they just choose not to breed for whatever reason, right? That would be a reproductive isolation. Would they be considered different species? In the zoo example, probably not, right? But in nature, it's hard to say. We also can't apply this to asexual species, right, that asexually reproduce, or species that are extinct, because we simply can't observe the environment that they're living in. This method a lot of times is used on plants, right? So we might use this to distinguish closely related species of modern plants. They might look similar, but they might be isolated from each other, so they have no chance of mating with one another. All right, we can use molecular features, right? So we can extract the DNA of an organism and compare maybe their entire genome, or maybe just a few genes, and see how similar is their DNA sequence, right? This is particularly useful in populations or in species where different species look similar. A lot of times algae and cyanobacteria look very similar to one another, but are actually different species. Right? In grad school, I took a class from a professor that this is what he did. He looked at these algae species that looked really similar to one another. He extracted their DNA, and based off their DNA, how different they were, he could tell species apart. Right? So we're looking at DNA sequences. We're looking at the order of the genes along the chromosome, the structure of the chromosome, how many chromosomes they have, right? Or are they tetraploid or diploid or triploid? Variety of molecular features. The drawback here is how different is different enough to be a new species. Right? So maybe 2 species of cyanobacteria, their DNA differs by 5%. Is that enough to say that they're a new species? Or is that just natural variation within a species? What if they vary by 10% or 2%? Where do you draw that line where a new species begins based on DNA alone? All right, our last characteristics that we are going to talk about would be ecological factors. Right? So this is how a species uses their environment. Right? So for example, these warblers up here might use their environment very differently. Right? They might use their environment different in the timing in which they breed. They might use the environment different in the food that they eat, or where they choose to nest. Are they ground nesters or do they nest in a tree? And those differences in environmental usage might determine if it's a new species or not. Right? They're using their environment so differently from another bird that looks similar, right, that their traits are different, that we could consider them different species. A lot of times bacteria are based on different species based on how they use their environment. Right? So do they grow on this substrate and not grow on another substrate? Even if they physically look the same, that might determine if they're a different bacterial species. Again, drawbacks, right? So different groups might have a wide variation of characteristics, right? So groups of bacteria sometimes display really similar growth patterns, even if they're the same species or different species. Okay. Birds, maybe it's hard to observe, right? They might look like they're using the environment differently, but maybe they have the ability to interbreed. Maybe that means they're one species instead of two. Right, so big picture is that there's so many different characteristics we could look at, right? It really depends on the species. What is appropriate for the species? Right, bacteria, it might be more appropriate to use DNA methods than, say, morphological traits. All right, so before we head out of class today, I want us to do this review. I want you to tell me the advantages and disadvantages of each trait, and we will do a review in 3-4 minutes here. I was amazed by how much faster this class is than the earlier class. Maybe I'm more tired in the morning at 8 a.m. and I talk slower. I don't know. Yeah, so this would be p squared is 1 phenotype, 2pq is 1 phenotype, 3 squared is 1 phenotype. So p squared, what letter should be assigned? Capital or the lowercase? The lowercase. Okay. So then squared would be lowercase This question so if 25% of the population is 400. Okay. All right, so you have 25%. Square root of 49% to get a singular. Yeah. That's then p would be 0.7. But now we don't have the square root of this one, because once we get the square root of 49, we get singular. Not necessarily, because I think we're giving comotypes and phenotypes.
What about it? Well, it's easy to tell like how different if you'll have like a bird and another bird that's kind of close in species but aren't the same.
I'm gonna review this let's do this on the board. Because I think writing it out instead of me verbally telling you it's gonna be
One of the disadvantages is
the same ones might.
Once we get into it, I just hear about that. It's a lot of we're going to talk about that on Thursdays. We're going to use it concept, which really relies on whether or not
yes.
All right, so we're looking at morphological traits to determine species, right? What is the advantage of using morphological traits? What's easy about it? Yeah? Yeah, it's easy to see. You can just look at the individuals. Is it gonna pop up? Oh, I think I have to click on my computer. Okay. There we go. It's easy to see. What's a disadvantage?
It could be completely different and be the same species.
Yeah, they could look completely different and be the same species, or they could look the same and be different species. It might not be so obvious, right? Also, you can look at how dissimilar do they have to be in order to be different species. Right? There's variation within a singular species. All right, reproductive isolation, what could be the advantage? What's easy about this method? What would you have to do to determine this method in a species?
See if they breed.
See if they breed, yeah. If they look similar, but they do not interbreed, we would call them a different species, right? If they look dissimilar, but they do interbreed, we would call them the same species. We just have to make observations on how these organisms mate, right? The disadvantage being? Yeah, it might not always be the case. It's hard to tell sometimes. Maybe they actually can breed, but they just choose not to for whatever reason. All right, molecular features. What's easy or what's the advantage about this?
Looking at DNA, you can see similarities really easily.
Yeah, we're looking at DNA, right? So we can detect different species if they look similar, right? So they might look similar, but when we extract their DNA, we can physically see that the nucleotide, the bases are different, or the amino acids that they code for are very different, right? The disadvantage being what? Yeah, drawing the line. How different does it have to be in order to be a different species? All right, ecological factors. What might be nice about this? What's the advantage? Ooh, this one's trickier, it seems.
It's easy to see patterns in the way that they, like their habits.
Yeah, we just have to, again, make observations and look at their habits, right? So if they use different habitats, we would assume that they're different species. But again, drawbacks. We're making a lot of assumptions here. We might not see all the variation in how they're using their habitats. We might not notice all the different ways they use their habitat. Right? So big picture here is there's a lot of ways to classify a species, and it's not one size fits all. Right? It depends on the species that you are looking at. Some might be more appropriate for bacteria, others might be more appropriate for mammals. All right, that's all I got for you all. I will see you again on Thursday. We will go over that problem.